Today I'll be presenting on an algorithm that we're developing in lab to reconstruct tumor phylogeny. And so before going into depth with what it actually is, I'll start with a brief review of metastatic cancer to kind of highlight the biological questions that we're trying to solve. Then I'll go through the basics of subclonal reconstruction. Before going through the method we're developing, we call it our algorithm because I haven't committed to a name yet. Then I'll show some results from that method and go into future directions. So for those of you who aren't familiar, cancer is a genetic disease characterized by dysregulation of cell proliferation, which can lead to the invasion and damage of surrounding tissues and organs. And so this can be broadly categorized based on the extent of the spread of the cancer into the following categories. So first, we have localized cancer, which is restricted to the primary site. And that means if you have lung cancer, you can only find tumors within the lung, and so on. Next, you have regional, which is where the tumor has spread to nearby lymph nodes, but no further than that. And lastly, we have distant or metastatic, which is when the cancer has spread to a different part of the body via the bloodstream or lymphatic system. And this figure on the right shows the relative survival for breast cancer, which, as you can see, drops significantly from 99 or 86% to 29% when the cancer becomes metastatic. And I think it's safe to say that this is true for most cancers. And a main reason for this is because definitive local therapy, which aims to remove cancer cells through surgery or radiation, is much more effective than systemic therapies such as chemo or immunotherapy. And so the reason for that is that heterogeneity between tumor populations or tumor cells plays a huge role in the efficacy of treatment that uses biomarkers to identify cancer cells. So as an example, if we have two tumors, one with only one clone versus another one with 3 clones. And here, a clone is defined as a group of cells derived from a single common ancestor, typically characterized by shared genetic mutations. And so when you have these two tumors and you apply the same treatment, in tumor number 1, where the population is relatively homogeneous, the treatment is effective in killing all the cancer cell populations. So this leads into remission. Whereas in the second population where heterogeneity exists, the treatment is able to kill the red cells, but not the purple or green cells. And over time, this cells population will grow, and this is when the patient have relapse. And so in addition to relapse, heterogeneity is also associated with metastases, because the more distinct populations you have, the more likely it is that one population have the ability to survive in a different site in the body. So the exact mechanism and pattern of this heterogeneity and how it relates to disease lethality, is a highly researched field. And so many studies focus on the genomic profile of the tumor at diagnosis. And this allows us to identify driver mutations that are associated with cancer incidence or relapse, but it doesn't really show us what happened before diagnosis and how or when the cancer came to be. Looking at tumor evolution focuses on reconstructing the evolutionary history of the tumor and inferring a phylogeny from which we can learn patterns of mutations that are selected for throughout the process. Walking through an example of how a tumor phylogeny would look like, first we start with normal cells. And some of these normal cells, mutations will accumulate, which results in higher rates of proliferation. And so these cells becomes the first cancer cell population, which we can call the most recent common ancestor, or MRCA. And as mutations keep accumulating, eventually some of these cells will gain a mutation that leads to an increase in fitness or even better cell proliferation, which leads to an emergence of new clones and populations, shown in like the purple and green cells. As time goes by, some clones might gain the ability to metastasize and colonize different tissues in the body. And this is when the cancer becomes incurable or leads to mortality. And so knowing the timeline of the development represented by the phylogenetic tree can help us identify the genetic changes that occur at each stage and how they might impact the effectiveness of cancer therapy. We can better understand what drives disease lethality and when is the most effective time to provide different types of intervention. To study tumor evolution, we need to first obtain the phylogenetic tree seen in the previous slides from sequencing data. Since most data are taken at or after diagnosis, this only gives us a snapshot of the current cancer cell populations. So we don't really have data on how the cancer started or what mutations represent that most recent common ancestor clone. And so inferring the tumor history through computational methods is called subclonal reconstruction. There are three main questions that we're trying to solve in subclonal reconstruction. And first is we want to look at the tumor composition of each tumor sample from a patient. And this means looking into the proportion of cancer versus normal cells in the sample, which is also called tumor purity, as well as the number of subclones and the proportion of each subclone within the sample. Next, we want to assign mutations to subclones to identify which mutations co-occur. And lastly, we want to reconstruct a phylogenetic tree by inferring the evolutionary relationships between each clone, all the way to the most recent common ancestor, which arise from the normal. And this helps us put the mutations into a chronological timeline to be able to study the process better, the tumor evolution process better. Today, I'll be focusing on the mutation profile and phylogeny inference. So the number of and composition of subclones can be resolved by clustering mutations based on their cancel cell fraction. And by cancel cell fraction, I mean the proportion of cancer cells in the sample that carries that mutation. And we can calculate that based on the allele frequency and tumor purity inferred from the sample. After we cluster the mutations, we can infer phylogenetic relationship between these clusters based on the average CCF of each clone. And so in this example here, where we have one sample and different mutations with different CCF values, we can cluster them into these three peaks. And based on this, we know that there's a group of mutation that has a CCF value of 1, a different group with a CCF value of about 40%, and another one with about 20% or 0.2 CCF values. And so based on that, we can reconstruct a tree that looks kind of like this. Most current tools in subclonal reconstruction uses a mixture of Dirichlet process and Monte Carlo Markov chain to cluster and build the phylogenetic tree using the mutations. So I won't go into the details of these methods, but some of the current methods that are commonly used includes PhyloWGS, and it uses a Dirichlet process, PhyloSub, and Pairtree. And so basically, these algorithm uses Monte Carlo Markov chains, or MCMC, to sample from the posterior distribution of clone trees. And it searches through the parameter space until it converges to the right solution. And so here, the right solution are the inferred subclones and the mutational profile, as well as their phylogenies. Since existing methods stochastically search through a parameter space, they can be computationally expensive. For example, some properties of Monte Carlo Markov chain includes flexibility. So since it allows you to sample from any posterior distribution, we are able to sample from this complex clone tree distribution. It also tolerates missing data and noise, which is great when working with genomic or biological data. It is also highly sensitive to the choice of models and priors, which is why all current methods uses the same Dirichlet process priors, because it has properties that capture the underlying biology really well. Lastly, when you use MCMC, convergence requires representative sampling of the posterior distribution. So what that means is that it needs to traverse enough of the parameter space in order to reach a good solution. And so this last point becomes extremely limiting when working with larger data sets, since the amount of sampling required grows exponentially with the dimensionality of the data. So they become infeasible when working with complex phylogenies, when you're working with multi-sample data. And so by multi-sample data, I'm saying patients with samples taken from multiple metastatic sites or across different time points. So we're proposing an alternative approach to subclonal reconstruction. The heuristic-based approach that we're proposing can significantly reduce the runtime and memory requirements for subclonal reconstruction, while hopefully maintaining a similar level of robustness and accuracy. So we can do this because we can leverage field knowledge, which, thanks to existing methods, we have a set of patterns and rules that are frequently observed through these unbiased methods and have been accepted as a community standard in subclonal reconstruction. So I'll go more into these specific rules in a couple slides. But then, since we're not iterating through a massive parameter space, the algorithm is faster since it doesn't require convergence and hopefully less memory intensive. So this overcomes the scalability issue we see in current methods. And lastly, since we rely on existing knowledge, the algorithm will likely struggle with cases that diverge from that. So another big part of our algorithm is that we also separate the clone identification and phylogeny inference step. And this means we're doing the clustering and the tree building in separate steps in order to simplify or restrict the parameter space for each step. So here's an oversimplified workflow of what the algorithm looks like. First, we start with the CCF values for each mutations. Then we run a clustering and filtering step in order to obtain the mutational profiles of each clone. In this process, we also discard some mutations that are not well clustered. And these usually are like low CCF mutations that look like noise. And the reason we do this is to simplify the problem further. And once we have the mutation profiles, we infer the phylogenetic relationships in order to build the tree. So going back to the heuristics or rules, instead of trying every possible tree topology and calculating the maximum likelihood, we simply use a set of rules to determine if a pair of clones can have different types of ancestral relationship. For example, clone A is a parent of clone B if for every sample that contain clone B, they also always contain clone A, and that in every sample, the average CCF of clone A is greater than clone B. And that is reasonable because if clone A descend from, or if clone B descend from A, that means every single cell that contains the mutation defined in clone B will also have the mutations that are defined in clone A. Next, we have clone A and B are siblings or parallel branches if there are at least two samples where in one sample the CCF of clone A is greater than clone B, while in the other the opposite is true. And this is called the crossing over rule in subclonal reconstruction. Lastly, we have clone A and B are the same clones if they're both present in the same sample and their CCF are approximately the same. And so this is typically a result of overclustering in the previous step. So everything should also fulfill the pigeonhole principle, where in each sample, the sum of CCF of the children subclones must be smaller than the CCF of their parent clones. And what that means is, actually, I'll explain in the next slide. Okay, so here's an example of a simple case which is reconstructed using our method. And as you can see in the right, the heat map represents the clustering results. Here we have one patient with 10 different tumor samples. And for each sample, we call the SNVs and calculate the CCF, which is represented by the colors in the heat map. And so here, the first two clusters represent mutations that are present across all samples at a relatively high CCF, whereas the ones on the right represents mutations that are present in only one of the samples. And so from that, we can build this phylogenetic tree on the right, which shows the relationship between these clones and the distance between them. And here, the distance means the number of distinct mutations it accumulated from relative to its parents. And so based on this analysis, we can infer which mutations appear early in the tumor, for example, in the MRCA or pink M08 cluster, and compare them to the ones that appear later, which are the ones that appear in these like leaf nodes. And when you run this analysis on a large cohort, it would allow us to identify mutations that are preferentially early, so always seen in the MRCA or early clones, and mutations that are observed in specific metastatic sites, such as the ones that are defined in the leaf nodes. And these are important for us to be able to target the tumor better, either in the early stage or understand how the metastatic tumors are different from each other. And so here's an example of a more complex case. And so here the clusters look a little bit more noisy, and it's not as obvious what the phylogenetic tree would look like. And this is where being able to implement the rules of subclonal reconstruction computationally becomes really valuable. And so this is what the resulting tree looks like. And again, this is way more complicated than the previous case. And it is also complicated to validate the actual results, which we're also working on. So since we're developing this algorithm to overcome the scalability issues in current methods, we benchmark it against Pairtree using simulated data. And we chose Pairtree because it is one of the only tool out there that is able to work with multi-sample reconstruction. So we simulated 576 SNV calls with different number of samples, different number of clones, different number of SNVs, and different read depths. And this is because we want to observe how the results and time and memory requirements changes with this various parameters. Okay, so here are some of the results from the benchmarking. Here are some box plots showing the distribution of runtime and memory usage. The y-axis is log scaled for visual clarity. And so I plotted these metrics for each sample. The red dots represent the results from our algorithm, whereas the blue one is from Pairtree. And so for each sample, I divide them based on the number of SNVs, number of samples, and number of clones. And based on these 3 plots or 6 plots, our algorithm has a faster runtime for up to 10,000 SNVs per sample up to 100 samples and 100 clones. However, the memory requirement seems to be comparable to Pairtree, especially for larger data sets. And we need to do more analysis to see if the memory complexity grows with even larger data sets. And lastly, we also group it by read depth, and it shows no direct effect on runtime or memory for both algorithm. So moving forward, we're going to benchmark the inferred phylogeny using the simulated data. And since we have ground truths of these phylogenetic trees, we will be able to measure the differences in accuracy and robustness comparing our algorithm to Pairtree. Next, we would also want to validate the algorithm on real whole genome and whole exome data sets. And this is to ensure that it is able to deal with the real life complexities we see in real data. So yeah, that's the end of my presentation. I would like to thank the Boutros Lab members and especially Paul and some of the people that are collaborating with me on this project. Thank you.